Mrs. Murdoch, and now we're going to actually do the measuring part of the Algae Lab. So in order to measure how the algae in these cups, like this one here, the algae in that cup is growing, what we're actually going to do is use a machine called a spectrophotometer, a spectrophotometer, long word. Here is a nice little explanation right here, if you can look at this, Mr. Bouchard. This is how you spell spectrophotometer. And inside this machine, what you have is a little lamp that shines on like a cuvette. This is a little baby test tube that we're going to put inside the machine. And the light shines through the test tube and then onto a sensor, which then measures the percent of light that's able to get through the test tube. So we're measuring something called light transmittance. So now that you have an idea of what's inside the machine and what it's going to do, let's actually do an example. So for example, if I put this clear water, all it is tap water, into the machine right now, and it measures the percent transmittance going through. Okay, well, it's a little bit high. This is my calibrating, okay? It went really high. Did you see the numbers went really high? We want the machine to think that clear water is 100% transmittance, which makes sense, right? Oops. Machine is a little finicky. We're going to go right to 100 if we can.
Sorry, guys. These machines are a little old, so bear with us a little.
Okay. So we use clear water to calibrate the machine. So ideally, when you turn the machine on, it goes right to 0 because it's waiting for data. And then if I put clear water in, the light, 100% of the light reaches the sensor, and it reads close to 100, and it does get a little fidgety. So right now it's calibrated, and that's good. So it thinks clear water is 100% transmittance, and it's at 0 when it's waiting for data. So now if I take the clear water out, let's just do a little experiment. This is water that I put methylene blue into, which is a very dark dye. So if I put this in here, what do you think it's going to measure? Think about that for a minute. What kind of percent transmittance would get through that very black water, right? So if I put it in here, oof, no percent transmittance at all, which we expected. And no light's getting through there at all because that's very, very dark water indeed, isn't it? So that makes sense. So now let's try, let me just try a little bit of, let's try a little bit of algae right from the tank. Right? As green as it can get. Nice dark, you know, algae. There is algae in sort of a climax community, right? At its maximum growth. It looks about like that. So if I put that in there, what do you think is going to happen to the percent transmittance versus the really dark water and the really clear water? It's kind of in the middle. So the percent transmittance, if you can guess, what's it going to be like? Let's see. Yeah, so about 45% of the light made it through to the sensor. So that's between 0, where no light made it through, and 100, where all the light made it through. So that's about what we're talking about. So in other words, the darker the water is, the lower the percent transmittance. The clearer the water is, the higher the percent transmittance. So as we are measuring our data today, we're going to make the first measurement, and I'm not going to do the whole thing because I just want to give you an idea of what we're doing. We already set up these cups where we put algae in the cups, and then we put some nitrogen of increasing amounts into each cup. Mr. Bouchard did that in the last video. So we have a cup that has 0 in it, 0 nitrogen in it. We have a 5 milliliter nitrogen cup, a 10 milliliter nitrogen cup, and 15 milliliter nitrogen cup. And right now, the amount of algae in each cup should be about equal because we used the exact same sample of algae to measure out each cup. So the only thing that's different in these cups is the amount of nitrogen in them. But we need our first measurement, our first percent transmittance measurement. So I'm going to take my first cup over here. I'm going to take my dropper and just put enough in there so there's enough for the light to pass through it and for it to give a reading on the sensor on the other side, because we know that's what's on the inside of the machine. So here's 0 milliliters. That's 0 there, so it's ready to go. It's calibrated. And it's gone up to 82% transmittance. So I have a data sheet, and I'm going to put 82% on that first 0 milliliter A cup. Now I'm going to take it back out of there and make sure I return the sample to the cup, and then return the cup to the light so it can keep growing. And then I'm going to keep going and do the next one. Let's see how close this is to that first one. Put some algae water in here and measure it again. One more. There we go. Okay. Calibrated. Go. Should be relatively the same. Yeah, it's right about the same. 82.8 is the beginning point for that one.
It went to 83.
83? All right, so 83%. Okay, 83%, that's fine. Back into the sample, back onto the windowsill. I'm gonna just keep going and get that first set together.
So guys, while she's doing this, think about what it is that's making it greener. What do you think is making this greener? Is it, oh, the water's just green, you know, it's polluted, that's why, you know. What?
And if it grows, what's going to happen to the percent transmittance if the algae grows? That's the other thing to wonder about.
These are pretty good.
Yeah, that's really good. Yeah.
We are brilliant with our data. If you guys just want to tell us how brilliant we are next time you see us online, please let us know. We are willing to accept praises of all sorts.
Okay, back in the windowsill. And the last one of that set is our 15 ml nitrogen and algae. So the question is, the more nitrogen you add, are you going to get more growth of algae? That's the question. To think about what your hypothesis will be. As you add more fertilizer, are you going to get more growth of algae? And if you do, is that going to make the water clearer or darker as the algae grows? Okay. All right. That's that first set. And I'm going to do the same with the others, but I don't need to have you watch me do that. So.
All right, guys, and we will be sure to take these measurements every day at the same time. So we'll probably do these right after school, same time every day. And notice we put them back in their same location.
Yep.
And same access to the outside, just to let you guys know.
Channel.
All right, guys, thank you.